The overall goal of this procedure is to examine changes in subcellular localization of proteins upon growth factor signaling. This is accomplished by first injecting animal blastomeres of Xenopus early embryos with RNAs encoding diversin RFP and the signaling receptor frizzled-8. Next, ectodermal explants and/or embryo cryosections are prepared and immunostained. Finally, the immunostained cells are imaged using a fluorescent microscope. Ultimately, results can be obtained showing how specific protein subcellular localization depends on embryonic signaling pathways. As an example, we demonstrate that diversin is recruited from the centrosome to the cell membrane by the frizzled-8 receptor.
This method allows researchers to visualize subcellular localization of signaling proteins in vivo using Xenopus embryos. This can help address fundamental questions in developmental and cell biology, such as studies of spatial regulation of signaling and cell polarity mechanisms.
To begin the process of in vitro fertilization, obtain eggs from female frogs that were injected with human chorionic gonadotropin 12 hours before the experiment. Add 0.5 to 1 milliliter of 1x Marks modified Ringer solution onto the eggs and fertilize them in vitro with a small piece of dissected testis. After 2 to 3 minutes, lower the salt concentration with 0.1x mmR to activate the motility of sperm.